Hi all, my name is William Garcia, and this is my presentation on spectroscopy in the human eyes. And let's go. So some uh, topics we're going to talk about is key definition spectroscopy related. We're going to talk about what is a wavelength, the light spectrum. We'll go in depth in the light spectrum. We're going to be talking about some key definitions eye related. So we're going to really dissect the anatomy of the eye, as well as spectroscopy and how both of them relate, which is what we talk about later. There's a dissection portion of this presentation. So after the key definitions I related, we'll have that dissection portion play. And then after the dissection, we'll talk about how spectroscopy is viewed in the human eye. So that's when we make that correlation between both. We'll talk about real world applications to optometry. So we'll talk about how eye doctors use spectroscopy in their daily lives when diagnosing patients. And then we'll talk about how my topic relates to our community. So to get started, some key definitions spectroscopy related. Spectroscopy is a study of the absorption of emission of light and other radiation by matter. Spectroscopy measures the frequency, energy, and wavelengths of light. Talking a little bit more about absorption. So absorption is the process by which light energy and light waves get drawn. Emission, the release of light energy. Radiation is the emission or transmission of energy in the form of waves. We're talking about light, so in the form of light waves. Matter is what an object is composed of, in our case, light. Radiant energy is energy transmitted in electromagnetic waves. Frequency is the number of cycles or oscillations that are completed in a measured way. Typically, we measure this in hertz. Wavelengths is the distance between 2 points on a wave from start to finish or left to right. A photon is a particle of light. The light spectrum refers to the different colors of light that exist, so UV light all the way to red light. And typically it's measured in nanometers to 400 nanometers all the way to 700 nanometers. So let's talk a little bit about what is a wavelength. A wavelength is, as you see from the picture, that is a wavelength that focuses on light. Wavelengths measure the distance in the full course of a light wave from left to right. This is in the same formula that we've used in class, where it's lambda equals V over F, which is lambda is the wavelength, V is the wave speed, and F is the wave's frequency. So the wavelength of visible light, like I said earlier, is from 400 nanometers all the way to 700 nanometers, or we could also see that as from red light all the way to violet light. So the visible light spectrum, on the far left of the color spectrum, we can see the red light, which is where the 740 to 625 nanometers are. And then, so that red light is considered infrared light, which is outside of visible light, meaning like we can't see that type of light. And then on the far right of the color spectrum, we see 435 to 380 nanometers, which is violet light or ultraviolet light, which is UV radiation. And with high exposure to UV radiation, it can cause skin cancer if you're obviously overexposed. As light increases to the right, the frequency and energy of that light also increases. As we go left, the wavelength increases. So as we can observe, red light has the highest wavelength, which is all the way from 700 nanometers, as opposed to violet light and indigo light, which has the lowest in terms of nanometers. So here are some more pictures that I have here, so you can see a better idea of the light spectrum. And now let's get into some key definitions eye related. So we're going to start with the cornea, which is a clear dome-shaped structure in the eye that helps bend light. The retina, which is a structure in the back of the eye that is light sensitive and absorbs light. The pupil is a hole in the eye where light passes through to reach the retina. Refract is the bending of light. So that's what typically is the cornea's function. Also the lens. Receptors, specialized structures that detect things. Photoreceptors are specialized cells that detect light and are commonly known as rods and cones and are found in the retina, which I'll talk about more during our dissection. Photopigments are light sensitive pigments that are in the photoreceptor cells. So they're inside the photoreceptors. Rods and cones are photoreceptors in the retina. So like I said earlier, photoreceptors are also known as rods and cones. The optic nerve is the nerve that connects the eyes to the brain. Macular degeneration is the loss of central vision. This is an eye disorder that is pretty common. Glaucoma is a disease that basically when pressure in the eye is too much to the point of blockages in the retina. So again, there's a whole slide on this later. I'll talk about it more in depth. Cataracts are when the lens becomes cloudy and causes blurred vision or blindness. So this is the dissection portion. I'm going to go ahead and let the video play so you guys can watch it. And I'll just go mute real quick. So
As we begin this cow eye dissection, we're going to make sure that we are suited up for the job. So we're going to wear a lab coat, some safety goggles, and some gloves as well, because we want to make sure that we are good to go for this lab. So as we start, we'll notice the exterior structures of the eye, which contain of the cornea, which is that right there in the middle, and the adipose tissue that's around. So with the cornea is basically the start of vision. It's where light goes into it to basically project light into the retina, which is the back part of the eye, which we'll notice in a bit. That right there is the optic nerve, which connects the brain to the eye. It's that little nub that you see right there. That structure is important because it basically sends electrical signals to the eye to create images. So now I'm going to be cutting into the eye. I want a front and a back side of the eye. So I'm going to be making a medial cut through the eye, through that sclera, which is the white part of the eye, and the fat along that. So to talk a little bit more about that cornea is the clear part of the eye. It would be clear, but since this animal is dead and it has been in chemicals, the cornea part of the eye is kind of blurry or, you know, foggy, which is normal due to the chemicals there. As you see now that there is some liquid coming out of the eye, that liquid is called aqueous humor, and it helps maintain the eye's shape, that sphere shape, and also the pressure inside the eye. We'll know that later on that that pressure that's in the eye is very important. We're going to talk about glaucoma, which is a disease that happens in the eye where there's too much pressure. We'll talk about that later on in my slides after the dissection. But to talk a little bit about what we're looking at from the outside of the eye, we're going to be talking a little bit about that fat that's around the eye, and I'll explain that right now. Then we'll talk a little bit about that adipose tissue, which is the fat that's around the eye. That fat basically keeps that eye safe from any movements that humans can make. For example, if I tilt my head, if that fat wasn't there, that eye would be banging up against that part of the skull, which will end up obviously damaging the eye. So if we didn't have that fat, we would be banging up our eyes all the time and causing eye damage, which is something we obviously don't want. As I continue to open up the eye, we're gonna be splitting the eye into two parts, and you're gonna be able to see some of the interior structures of the eye. Okay, so that is the anterior side of the eye, or the front side of the eye. So you're going to see this structure that looks like little ridges around the eye. Those ridges are called the ciliary body. They're basically muscles that help constrict or relax the pupil. The pupil is the hole in the eye that allows light to go inside, which is that hole that we see right there in the center. So yeah, those muscles basically in highlight settings, they will constrict because they don't need as much light since there's already a lot of light in the room, and in darkness the pupil expands, there's less light, so the pupil needs to be bigger to get that light. That black part of the eye right there without the ridges is called the iris, and that is what gives our eye its color. And the back side of the eye, you're going to notice that ball-like structure, which is the lens. That is basically what projects light to the back of the eye. It pinpoints it back to the retina, which the retina is under the lens. I'll take the lens out right now so that you're able to see the retina. You're also going to notice some gel-like liquid or substance that is called vitreous humor, and it acts the same as aqueous humor, which it just makes it gives the eye its shape, and it helps maintain pressure inside the eye as well. So what we're gonna see now is the retina, which the retina is supposed to be all flattened out, but the way I cut it, I wasn't too gentle with it, so it kind of clumped up right there in the center. But that retina is where all those photoreceptors are, and that's where we could see that spectroscopy take place. The retina absorbs the light and creates it into electrical signals, and there's a pinpointed side to the retina, which I'm going to move the retina around a little bit so you can see that. But inside the retina, there's photoreceptors called rods and cones, and also photopigments. Now that I moved it, you could see that there's a little spot in the retina where it connects. That little spot right there is where the optic nerve gets connected to, which is again that nerve that uh, turns it in. Yeah, right there. That nerve that turns it into electrical signals and sends it to the brain. So I'm gonna peel that retina up a little bit so that we can see that beautiful blue color part of the eye, which is a part of the eye that only animals have. Again, this is a cow eye, so that part of the eye is basically what helps animals see better at night. A lot of nocturnal animals have this, but it's pretty common in most animals. It's called the tapetum. I'm going to peel the tapetum right now so that we can see that back part of the eye, which is the vascular part of the eye. It's called the choroid. Once I get this peeled up, you're going to be able to see that nice black color. You can kind of start seeing it right now. That part is basically what gives the eye its nutrients. It also helps the retina out by absorbing that light. Again, that tapetum layer is what animals have, so our eyes would only have the choroid in the back. So that choroid absorbs light as well, and it's highly vascular, meaning it contains a high amount of blood. On a live organism eye, that black part would be pink or red to show, that it is vascular. That right there is the retina again. Just to, I guess, reiterate, rods and cones, they basically help us out by helping us determine different colors. It also helps by helping us determine shading, shapes, and other things like that. Without rods and cones, we would not be able to see colors. So again, that's the lens right there. The lens would be clear originally, but since this animal has been dead and it was in chemicals, it is now a yellowish color. That lens, when it becomes like that color right there, yellow, that's when someone has cataracts, which I'll explain later in some slides. But it's important to know that that lens would be clear if it's a living organism like us humans. Again, the posterior side of the eye, choroid. Oh, that's also the sclera, the tough layer that's around the eye where I cut in originally. That's the sclera. We all have the sclera, the white part of the eye. Something that I'll delve further into in these slides that are going to be up ahead is some eye disorders or diseases, for example, glaucoma, cataracts, and macular degeneration. I'll be going over that shortly after this dissection. So the last few takeaways to take into account is the lens being an important structure, the cornea, the retina, photoreceptors, photopigments, and the pupil is also very important. These are all very important structures that I will be explaining in detail in the next few slides. So this has been the dissection segment of this video. Again, just a reminder, I will be explaining things more in detail in the slides that follow right now. Thank you.
Spectroscopy, right? How is spectroscopy viewed in the human eye? So when light enters the eye, the light rays are bent by the cornea, which is that structure that I showed, the clear structure in the front of the eye, which is also here in this picture. So the light rays are bent by the cornea, and it helps refract the light to the back where the retina is, that pinpointed location. So the light then passes through the pupil and it reaches the retina, which is in the back of the eye, where light is absorbed. Referring back to light wavelengths, and according to Anne Marie Hamstein, the eyes can detect wavelengths from 400 nanometers all the way to 700 nanometers, which is what we've been seeing. This is from UV light to IR light, which is infrared light. Um, so the retina uses photoreceptors such as rods and cones to send electrical signals to the brain through the optic nerve, which is part of the brain that connects um, the eyes to the brain, which ultimately creates the images that we see every day, right? So you're literally using it right now as you're seeing this presentation, because if it wasn't for these rods and cones, if it wasn't for this brainstem, our eyes wouldn't be able to see the things we see every day. So chemically, rods and cones are also called photoreceptors, which are specialized cells that receive light from the retina. Inside those photoreceptors are photopigments, which are used to absorb the light. The photopigments go through chemical changes in low light settings as well as highlight settings as they constantly adapt by changing their shapes. So, um, according to vision transduction of light, when light hits a photoreceptor, it causes a shape change in the retinal, altering its structure from a bent cis isomer and to form of the molecule to its linear trans isomer. This means that photopigments and photoreceptors are going through constant chemical changes. So we could see these isomers right here. It's the cis retinal and then the trans retinal. So real world applications to optometry. Optometry is the field of study and profession in healthcare of the eyes and diagnosing visual defects and prescribing help through glasses or corrective lenses. Spectroscopy is crucial to the optometric field as it is used to diagnose disorders and diseases like blindness, macular degeneration, glaucoma, and cataracts. These are just a few. There's obviously a ton of diseases and eye disorders, but these are some of the most common ones and some I'll be talking about. So spectroscopy can also help explain the wavelengths of light and how humans are able to detect as many wavelengths are invisible to us, because obviously there is light, sorry, that we can't see as humans, but there's also light that we can see, which is the light we've been talking about. So this is an example of macular degeneration. As you can see, macular degeneration, there's like a spot in the center where the vision's blocked. That is due to blockages of the retina where light doesn't fully get transferred back all the way back to the eye. Here's an example of glaucoma. Glaucoma is when that pressure buildup happens and then the eye becomes red and swollen. In many cases, this requires surgery, and it's scary to have because this isn't a disease. This isn't a disorder, sorry, it's a disease. And then here's an example of cataracts. It's when the lens becomes cloudy. It's either the lens or the cornea that becomes cloudy. Either two can happen. This also requires cataract removal surgery, which is this is probably the most common eye disorder that you can probably get. Many people, this is pretty common. This is one of the most common forms of blindness. The cornea or the lens becomes so cloudy to the point that vision just becomes really blurry. So my topic and its relation to our community. So obviously this topic is pretty broad. So it's not just my home community that it relates to, it's everywhere. It relates to you, it relates to me, it relates to my family, your family, everybody in the world, because we use it every day. Without our vision, our lives would be vastly different, which is true because obviously every day that we wake up, every day that we sleep, from the morning till nighttime, we are using our eyes to see things, right? So day after day, every time we use our eyes, we are using chemistry. In fact, again, you're using it right now to see my presentation slides. So thank you for watching. This has been my presentation on spectroscopy and its relation to the human eye. Here are my works cited page. The images that I've used were cited throughout the slides. So yeah, thank you for watching. This has been fun to do, and